title is over there.
Yeah. So thanks a lot for inviting me to this very wonderful place and to this very interesting conference. And I will speak about a work basically done by Jérôme Toubiana under the supervision of Rémi Monasson and myself on applying neural network and in particular restricted Boltzmann machine to understand data, in particular protein sequence data, and I will describe the statistical physics of learning of these machines. So basically the idea is that we'll deal here with unsupervised learning in the sense that we have some data which are non-labeled, but we know that they are good configuration, so maxima of a probability distribution. And what we want to do is to infer this probability distribution from the data. And I will try to show you that this problem has many applications to model complex systems where first principle approaches have limited success, and the key application will be to understanding the relationship between sequence and structure and function in protein data. And I will focus on an unsupervised neural networks, which is called a restricted Boltzmann machine. And the interesting point I will underline is that once you have inferred from data this probability distribution, of course you can sample it to generate new configuration which are good configuration for the system. So data, so I will describe three kinds of application. The first application is the very well-known benchmark data set on machine learning, which is the MNIST digit data set. So this is a set of 70,000 binary images of handwritten digits, number, and you can discretize them in 28 by 28 pixel. So you have a binary variable in which you have 0 or 1 corresponding to the fact that the pixel is white or black. And another data set on which I will focus are protein sequences, because now it's possible to sequence a lot of genome, and the sequences corresponding to the same protein are collected on database, biological database, which are, for example, called PFAM. And this is an example of sequences which all fold and function on this small protein domain, which has 31 sites, as which is called WW domain, because it has these two W, which are very important for the protein. And in this case, like in this case, you have configuration of bits. So each possible amino acids here, so it's possible of 31 sites, can have just 21 possible value. So this is a generalization of an Ising model of binary variable to Potts variable, which are categorical variable. So they are described here by letters, and we have 21 possible letters in this case. And we have also benchmarked our approaches to many artificial data. For example, we show the example of data from one de-Ising model. So these are 100 spins, so these are configuration of 100 spins. There are 10,000 configuration of this system, and again, the variables are binary, so they are black if the spin is up and white if the spin are down. And again, we'll infer from this data a model. And so why we want to learn from this data a model or a probability distribution? Because we want to understand the structure of the data. We want to make prediction in the sense that if we have a model, then, for example, for family sequences of protein, if you give me a new sequence or a mutated sequence with respect to the one which is known, I can give you the score, so the probability that this new sequence is a good sequence for this protein. And of course, also, we, as I have said before, we want to use this probability distribution inferred from data to generate new artificial samples. And again, in the case of protein data, this will be used to design new protein, so new protein sequences, which can be experimentally synthesized and tested. And so the architecture in which I will focus is this architecture, which is called the restricted Boltzmann machine. So it's a case in which the units, input units, which are here, and these are the visible layer. For example, these are the n sites of your Ising unidimensional model, or the n amino acids of your protein, and these are connected to a weights vector to a layer of variable, which is called hidden layer. So this is a very shallow deep network in the sense that it has only one layer. And we started to analyze this architecture because it's simple, and to try to understand something about this, what this the meaning of this weights vector. And I will show you that this machine are able to explain data through their common feature, and one can combine feature to in turn generate new data. And what we want to understand, for example, typically on MNIST sample, we want to understand what is the difference between these G digits and random binary images. So the first point you can immediately see is that there are more black pixel than white pixel. And if you sample a random sparse binary images, this is what you will have. So you are not obtained yet at a stage to reproduce this number, but you can also see that white pixels are grouped in strokes. So if you sample random strokes images, you see something, you start to see something, but there is more, the fact that the strokes are arranged in a specific way, and this is what RBM is able to capture from the data. And so the artificial sample generated by RBM look really like real sample from MNIST. So this is the machine I will explain in the next of the talk. And the other big application I will underline is on proteins. So the idea is the proteins are sequences, are just a strand of amino acids which are folded in a very specific way and which have a specific function. For example, this WW domain has to bind some ligand and to signal its binding. So there are a lot of constraints which are encoded on this sequence, and it's very difficult from first principle methods, for example, from simulation and folding simulation, from the knowledge of all atomic potential between all the atoms to guess the structure from the sequence. But and there is more, so the sequence doesn't only fold in one or few native folds, but it has also to bind the target ligand in a specific way, and it has often a catalytic activity, so it must promote reaction within the target ligand. So we want to see how all these constraints are encoded on the sequence. And the fact that now we have a lot of sequences, not only just one, so we can use all this data to infer this constraint. And for example, this is the multi-sequence alignment for this, a part of the sequences on the multi-sequence alignment for WW domain. And we would like to understand on the data why these sequences, which works, fold, and function correctly, are different from sequences which look similar, but they are not functional, as it has been experimentally tested. And for example, one first things one can say is that I will see just the frequencies of the different amino acids and on different sites, but this is not the only as the white-black pixel in the example of MNIST, but in this case, for example, the two sequences are quite different. They have only 50% sequence identity, but they have the same activity in vitro. And on the contrary, these two sequences, which differs only from one amino acids, one is functional and one is not functional. So we would really to understand the difference between, able to infer from data the difference between these data. And the second important point is that, so this, as I have told you, these sequences are sampled from different organisms, and they are clustered in the space of possible configuration. So one can, the idea is to extract from this data also the different cluster correspond to different subfamilies of this family, family. For example, subfamilies in which the specificity through the ligand is a little bit different. And this is what I will call a feature extraction from the data. And this is typically done, for example, by principal component analysis, in the sense that one can take the correlation matrix between the occurrence of the different amino acids in different sites, diagonalize it, and you will find some principal components or principal modes. And then the sequences can be projected on this space to characterize these maxima of the probability distribution. And this has been already done by several people in the field of biophysics and analysis of protein sequences to find which are, for example, the position which are important for the different specificity, for the specificity of the protein. But this principal component analysis is not generative, so we will extend this concept of extracting feature to a machine which is also able to generate new sequences, and this is what RBM does. So in the case of principal component analysis, they just extract the few 1, 2, 3 principal component. And I will show in the case of RBM, in putting sparsity and so on, you extract a number of principal components, which is much larger of the order of the number of sites in the protein. But you have really to be careful to infer in a proper way these weights, for example, in the presence of few samples, so in the presence of noise. And so I have, for example, sparsity constraints and so on. So this is what I will discuss. Okay, so what is the idea? Is to going from the data space. So data space is a very high dimensional space. For example, for binary images, this is 2 bytes to the power 76. And in the case of binary of protein, this is a larger protein space because there are 20 amino acids for each site, so it's a space of dimension 20 to the power n. So sequences, again, can be represented by points on this multidimensional space. And these are all the functional sequences for a given protein, or, for example, all the set of images of digits. And what we would like to do is to project this space in a lower dimensional space, which is the space of the feature. And this is exactly the space of the hidden units. So there are as many features we want to extract from this data as hidden units on this restricted Boltzmann machine. And for example, the idea that each of these features will extract something about your data. For example, the strokes in the first example of images, or protein biochemical properties like the specificity, the activity, and so on. And so you will cluster in this space of sequences into this different cluster, which will activate your hidden units in different way. And so the idea is to go from this space to the hidden space by a probability distribution, which is the probability of the activity of the hidden units given an input sequence. And then you want to go back in the sense that you want to define a probability of generate some data or some sequences having fixed the hidden unit values, and this is the probability of your data set given your hidden unit configuration. And you can even generate a new configuration or new sequences which have not been experimentally observed. And so this is done by this restricted Boltzmann machine. So they are a graph, bipartite graph of a layer of visible units. And these visible units can be, for example, binary in the case of images, digits. And they have a pot variable where each variable takes the 21 possible letters corresponding to the amino acids in protein for protein sequence data. And then you have this hidden layer variable with M hidden layer, and you have the 3 terms which are described here in the potential. So the probability, joint probability of having a set of variable here and hidden variable here is just has an exponential weight with an energy which has 3 contributions. So local fields terms on the visible units, Then there is potential terms on the hidden units, which is analogous to this local field terms on the visible units, and the couplings between terms, which is this weight matrices, between the variable, visible variable, and the hidden unit. And so, for example, if you want, first of all, given a configuration, you can calculate the input that the hidden variable receives as the projection of the configuration on the weight vector, and this is these terms. And once you have this hidden, this input, the probability that the hidden layer is activated with this value of the hidden variable, given the input configuration, is just the exponential of minus this way, these potential terms, plus the value of the hidden unit times the input. So the projection of the visible configuration on the hidden layer determines this input to the hidden variable. So for each hidden variable, you will have a input which depends on this weight vector, and this in turn determines the probability of activation of this hidden unit. And then you can also fix the hidden unit and sample a new configuration, and in this case, the probability of, yes? Just because this is the exponential of minus the energy, and the energy has a sign minus. This is just a notation. You can put a plus or minus, then you will have terms. Yeah, you had a question also? Okay. And so you can, so when you fix the hidden units, you can then sample the visible units just by, in this case, the probability of the visible units given an hidden units configuration is just the exponential of these field terms, plus again the projection of this weight on the and the hidden units value times the hidden units. So this is or realize exactly what I have described here. So you can, you can have both the probability of activating a hidden unit given a visible configuration, and the probability of generating a visible configuration given a hidden units configuration. And then, so how you learn these parameters? So parameters basically are these weights parameters here, and these input fields. And you learn this parameter just by integrating this probability, joint probability distribution over the value of all possible configuration of the hidden units, and this gives you a probability to obtain a given sets of visible units, and you maximize the probability of your data, so your observed sets of visible units given your parameter, and in such a way you find the parameter which maximizes this distribution. So RBM are generative models, and they are trained through unsupervised learning to and this is done by finding the parameter which maximizes the likelihood, this probability of your data given the model parameter. So this is a difficult problem because there are a very large number of parameters. Yeah. Yeah. Now these are, yeah, there are both function and parameters. So this is what I'm describing. So the parameter, the weights, so the weights are just parameters, and also these field steps are parameters. They are function in the sense that takes 20 or 21 possible values depending on the amino acids here for a Potts model. For the case of a Ising model, you will have just a local fields parameter with one value. In this case, you have a local fields vector which has 21 values corresponding to the different Potts state, which are the different amino acids. So this is what I call parameters. It's a function in reality. But you find all these local, you have to infer all these local fields for every position and every amino acids. And there, these are these other vectors of couplings, or what we call often weights, between these variables and hidden variables. So for each hidden variable, we have again a set of parameters which depends on the position and on the amino acids. But then, so these are the easy parameter in the sense that are the one which will be fixed through maximum of the likelihood. But there are also other hyperparameters of the network. For example, the number of hidden units. And so this is related to your question, how many modes you want to extract from your data, or how many features you want to extract from your data, and the shape of this potential. And we'll see in the next that these are very important parameters to fix in an appropriate way if we want to have an interpretable and well-working machine. And also the sparsity on this weight vector may be very important in the case of limited data. So, and how we can, once we have fixed the hyperparameter of the network, we'll infer these fields and weight parameter by stochastic gradient ascent of the log likelihood. And this is also a difficult problem because it's not guaranteed to converge to a unique solution in this case. And so let's deep a little bit into the hidden unit potential shape. So the simplest hidden unit potential shape is the Gaussian potential. So in this case, this potential is quadratic, and one maximize with a quadratic term here the probability of activation as a function of an input. The most probable value of the hidden unit given the input is a linear function. So these are called linear RBM. And this is a very interesting case, and probably, yes, I will not go into the detail, but probably Mark will explain a little bit this better. But you can, when you integrate this probability with a overall possible value of the hidden unit with this Gaussian potential, you will find a network model which is an off-field model which has M patterns. So the patterns in this case are the minima or the maxima of the minimal of the energy or the maximal of the probability distribution I've sketched before. So a linear RBM is just a network in which everyone is connected and in which every visible unit is interconnected, and in which the couplings parameter of interconnection are described by low rank metrics. And another potential is called the rectified linear unit. It's like the linear potential, but the hidden units will activate only if the input is larger than a given threshold. And this potential corresponds to a potential which has at the beginning is a linear with a slope theta, and then is quadratic. And this is interesting because it generates a lot of effective multi-body interaction between the visible units when you do the integration over the hidden unit variable configuration. And another kind of potential which is used in this kind of, network is Bernoulli-like potential, in which the hidden unit have discrete value, and for example, they can be 0 or 1, and this corresponds to a transfer function of this type. And there is a work done by Jérôme and Rémy in which they have studied the statistical physics of learning, and they have seen that depending on the hyperparameter and on the potential you put on your RBM, there are different data representational phases which are separated by true phase transition. And there are phases in which the learning is easier for this network, and you have more interpretable results for the weights. And in particular, this phase is called, they have called this phase of good learning a compositional phase, and which give better results in the sense that the training and sampling are easier. So practically what you have, for example, as a function of the, here is the function of the value of the potential on the input units, you have that for very negative values, you have a prototype-like representation in which, for example, in the case MNIST, a configuration for a 5 will activate only one hidden unit, which will recognize a prototype of this 5. But on the contrary, in a compositional phase, a 5 will activate different hidden units corresponding to the different strokes of your 5. And so this is why this is called compositional. And then there is another phase which is called glassed phases or entitled, in which every input configuration will activate all the hidden unit values in the hidden unit in a weak way. Yes. Yeah, exactly. For example, quadratic potential doesn't have this compositional phase, but only Bernoulli-like potential and ReLU potential, the ReLU activation function have this composition phase. So they have studied exactly how the potential, the number of hidden units, and all the parameters gives the presence or not of this compositional phase. And to give you an example of the fact that the linear RBM doesn't work as well as ReLU RBM, for example, in the case of M-list, you see that what you generate after training an RBM, a linear RBM or a Gaussian potential on this data are numbers like this, so they are not very nice. And you see that sometimes you stuck into some configuration for a very long time. On the contrary, if you have ReLU, you will generate digits which look very much similar to this data set, and you make transition more easily between one digit to the other, and this is thanks to this compositional phase, because you just turn off, for example, an edge, and to on, for example, you go from a 3 to the 8, just turning off 2 edges, and so on. So it's very important for the learning and for the representation to have this kind of compositional phase.
On a more theoretical level, understood what feature ReLU does this?
Yeah, because in more theoretical understanding, they have studied really the energy, the free energy of the problem, and seen that there are phase transitions. So, for example, you can have several minima with ReLU that you don't have with the Gaussian potential. And so I will, and so for the case of protein sequence data, it's even a little bit more complicated in the sense that to well fit the data in the presence, which are a few data with respect to the number of parameters, so it's in the presence of undersampling, we have a fixed adaptive nonlinearity in the sense that you have a potential which is a priori a double ReLU, so it has 4 parameters, 2 slope in the beginning, 2 curvature parameter, and you fit also this parameter through learning, so you can go from, for example, quadratic hidden units to ReLU hidden units or Bernoulli hidden units. You can adapt your hidden units to your data. And also a very key important fact is to adapt, to impose sparsity through regularization, and we'll see it better in the next. So I will try to show you in the next that in this compositional phase, weights are interpretable. So this is the key factor of this very simple machine, that you can really interpret these weight vectors. And I will show it into 3 examples: the M-list set, 1D Ising model, and protein data sequences. So first, yes?
This comment on the PCA. The big
Yeah.
thing on PCA that you can, it's not a cut, but you know where to cut.
Exactly.
You look at the eigendynamic,
Exactly.
you cut.
Yeah.
Here, how do you choose the size?
Yeah. Yeah. So all these hyperparameters at the end are chosen by cross-validation. So I will show you just the result, for example, on the protein data sets to show you the example, sorry, if I find it. Yeah, it's at the end of the talk, but this is the idea. So don't look at this, but this is the idea of the cross-validation. So, here is cross-validation as a function of the penalty strength, but you can do the same things as a function of the number of hidden units. So the idea is, and this is what classically one does in machine learning. So the idea is one fix the hidden units, the number of hidden units, your hyperparameter, and then one separate the data into set, a training set and a test set, and one infer the log likelihood of the training set, which is these curves, and one compare with the log likelihood of the test sets as a function of your hyperparameter. And typically, you put yourself at the value of the hyperparameter where the two log likelihood are close enough. So here at the beginning, you are overfitting your data, and the end, for example, in this case, we see that the penalty strength is too large, but here you obtain a good log likelihood on the test sets with a minimal possible value of this penalty strength. So this is exactly what we do for the number of hidden units. So it's very more involved than the principal component analysis, but it's really related to the same idea. So depending on the sampling, I will choose my hyperparameter, my number of hidden units, and so on. And how many data, sampling in the sense how many data I have. So going back to weights, so I will try to convince you that these weights are really interpretable. So let's start by the weights you obtain on the MNIST data sets. So these are all possible, the weights for the all possible digits. And you see for several hidden units, and you see that when you have few hidden units and you are in this prototype-like configuration, weights resemble much to the single digits. So these are the 16 weights one infer when one just has here 16 weights. When one put here a much larger number of weights of the order of, I would say 50 here, you will have that weights start to resemble to the strokes of the images. So then you can, when you activate different hidden units, you compose a digit by the composition of these weights. And another interesting application, which is very simple, is what's happened in to this weight in the case of configuration sampled from 1D icing model with boundary condition. So these are the data I have presented at the beginning. So here you have 100 spin and 10,000 sample configuration, and you will have something like this. And you want, so this is how you have generated the data. And this is work done by Moshir Arsh during internship stage. And so we have started by learning RBM on this system with just one weight to see what the weight in this case will look like. And so we have, during learning, we have measured log likelihood, and here I will show you the weight you have, for example, here, when you have reached a maximum of the log likelihood. And you have that this weight is focused on some sites, so on a limited portion of your ring. So it's just looking at some spins here and ignoring the other. And why it's looking at this group of spins? Because this group of spins correspond to a group of spins with the length, so has a size of the correlation length of the system. So to infer, in this case, the physical characteristic, for example, the magnetization, you need only to look at a group of the spin which correspond to the correlation length of the system. And so, this is these weights, exactly these weights. And then, said in another way, the good sequences are the ones which are up, or the spin up, or all the spin down on the correlation length of the system, and you don't care about other region, you want to be aligned on this correlation size. And when you change temperature, you change this bump width according to the change of the correlation length. So you are inferring the meaningful scale at which to look at the system. When you you want to see how these weights are made for different value of log likelihood, so you continue learning and you keep the log likelihood of this plateau, you will see that these weights will diffuse, so the position of the weights will diffuse, so you will see all the system. So you will, in some sense, restoring the dynamical symmetry of the system. And if you put multiple hidden units, you will have a different bump corresponding to the different region of your data set of your ring here. So each hidden unit look at the portion of the system corresponding to the correlation length of the system. And this, so while the first case of one only hidden unit was corresponding to the mother cell representation, in which only configuration which are aligned here will activate the spin, here you restore with multiple units the compositional representation in which each unit look at the portion of the space. And this is interesting because it's much like what happens in the brain. So there are a lot of sets which respond to precise, to stimuli in precise region of the space. For example, place fields in the hippocampus. So next, I will show you what weights look like on when inferred from protein sequence data. So again, this is a multi-sequence alignment containing some sequences of the WW domain. So there are much more, like 10,000 of such sequences. And the first thing you can notice that there are some amino acids which are really conserved. And this typically in biology, bioinformatics, is represented by this representation, which is called the log representation, in which the height of each symbol here corresponding to the amino acids correspond to its conservation, so to its probability to be observed in the amino acids. So the first feature, statistical feature you can see are the presence of these very conserved sequences, very conserved amino acids at some position, and this is extracted by the frequencies of amino acids at each position. But there are also some correlation you can extract from the multi-sequence alignment. For example, between these two sites, here and here, where the sites are more, the amino acids are more variable, you will have that often when there is amino acids, so here the color are the type of amino acids, and here are amino acids which are charged in a negative way and amino acids which are charged in a positive way. So these two sites that are indeed in contact in the three-dimensional structure of the protein show correlation in the sense that if one site is charged negatively, here the other must be charged positively, or sometimes these are tiny amino acids on the one side which are correlated in the multi-sequence alignment with these negative charged amino acids. And from the inference, direct inference of network, it has been shown that, especially by starting by the work of Martin White, and this is a review paper, that this, when you infer a direct network between these amino acids, you can indeed find structural information, so the amino acids which are most tightly coupled correspond to amino acids which are in contact in the three-dimensional structure of the protein. And this has been very successful to solve, in some sense, or going through the solving of the protein folding problem. So what weights look like? So there are a lot of weights which look like contacts. So, for example, this is one hidden unit, and this is the vector of weights which is connected to these hidden units, so weights number 1. And we have adopted the same log representation for the weights in the sense that weights can be positive or negative, and the height here of the letter correspond to the entry of the weights corresponding to these amino acids. And you can again see that, for example, these weights will have negative inputs for all sequences containing the pair of negatively and positively charged amino acids on these two positions, and will have a positive input. So will, for example, activate or deactivate the hidden units depending on the fact that sequences are like this or sequences have these patterns of correlation. So these are a lot of weights which are localized and correspond to the part of small eigenvalues, for example, in the correlation matrix, which is also very important for structural prediction. But then there are other weights which are less localized, and these, for example, are related to binding specificity. And WW is known to have different binding specificity, so it prefers, depending on which sequence is here, the protein will prefer to bind ligand which have different amino acids composition. They are basically all rich in proline, but there is one amino acid which is different normally on this ligand. And this defines several clusters or several subfamilies in your sequence space, which are called group 1, group 2, group 3, or group 4 of sequences. And the preferential binding towards this ligand has been experimentally verified. So principal component analysis and a version which has been much more introduced by the group of Ranganathan, Stein, Leibler, which is called the sector analysis, identify that there are like 8 positions here and here, which are very important in these modes, so they can, in some sense, isolate this group of amino acids which are important for the binding specificity. And when you do, when we have trained our RBM on this data set, we find back something which is very similar to this vector. So these are more or less the 8 positions which were identified by this sector analysis, and this was just one weight vector, the principal component weight vector. And these are, for example, the weights attached to another hidden units, the hidden units number 3. And one, for example, key amino acids for binding ligand of type 1 is this histidine as this position, and you can see that indeed this is very important on the weight vectors. So you see really the pattern here of the sequences which bind as type 1. So this is the important amino acids, and you can see the letter here correspond exactly to this pattern. So this group of sequences correspond to type 1 sequences, and all type 1 sequences will have a negative input on the weight 3, and the other sequences of type 2, 3, and 4 will have this pattern on these amino acids. But for example, there is another weight vector which we call 4, which is localized on this loop here, and is important to identify sequences of group 4 with respect to sequences of group 1, 2, and 3. So practically, you can, by the input, classify the different clusters corresponding to the different type of sequences and type of specificity. So, yeah. No, it was just one vector which had 8 component, weight important component. And this, in some sense, was able just to, and this was the input 3, in some sense, this was the projection. This vector will give you just the distinction between type 1 sequences and the other, but you can do better, for example, in this case with another component which we found with another weights. You really clusterize more or less the 4 type of amino acids. There is the type 2 and 3, which are quite of sequences, type 2 and 3, which are a little bit still together, difficult to distinguish. And then there are the unknown types. So these are all sequences which have been experimentally tested with respect to the binding properties. No, with the PCA. Yeah, exactly. You did, yeah, exactly. So you have a better identification of these clusters. And so next, and this is the other important point, and I will more or less finish with this, is the sparsity. So it's important to introduce sparsity to have a well-interpretable vector. So typically, this is the value of penalty strength. So we have enforced the sparsity through a L1 square penalty, meaning that we want sparse weights, but we want that the norms on the different weights be more or less similar. So here is what you obtain when you are at this value of the penalty strength. Of course, if you are at this value at the penalty strength, you are overfitting the data and you cannot see anything, so you cannot see your pattern. And if you are at a smaller penalty strength in which you have really the maximum here, also you don't have very nice and interpretable weight vectors. So this is what we call the interpretable interpretability-performance trade-off. So we are in a value of log likelihood for the training test, which is for the test set, which is a little bit smaller, but which gives you weights which can be interpreted. And yes? Yeah, but then you have something which is too sparse in some sense. It can be too sparse and have a little bit lesser log likelihood. So you lose a little bit in log likelihood. But yes, so there is, in some sense, the idea is that moving on this penalty strengths, you will have a sparse and sparser weight, and you will lose something in log likelihood, but less here, for example, it will not change so much, even if you go to this value. And this is what you have with this cross-validation analysis. So lastly, you can generate sequences by your RBM, and so these are all sequences, for example, generated by a Monte Carlo simulation of your probability distribution, and you see that it correctly clusterized on these three clusters of sequences we had identified before, but you can do more, so you can fix the hidden units to sample, to generate artificial sequence only on one specificity class. For example, in this case, you fix the hidden unit 4 to a positive value, the hidden unit 3 to a negative value, and you will obtain sequences of type 1 specificity. If you, on the contrary, fix the 2 hidden unit to positive value, you will have type 2 or 3 sequence specificity. And if you fix the 3 to a positive value and the 4 to a negative value, you will have a type 4 specificity sequence. And you can also fulfill some region of the space which have not been sampled. For example, fixing the 2 hidden variable to a positive value. And in this case, you will obtain sequences which are here. So I think, yeah, I have to stop. So as a conclusion, I've shown you that under specific conditions, which are weight sparsity, non-linearity, the RBM learn a compositional representation of data, and they achieve a good trade-off between interpretability and performance, and they can be used to extract meaningful features from sequences data. For example, the type of specificity of the sequence, and then they can use to generate sequences even with specific properties, again with specific specificity. But RBM are less well-known and studied model than, for example, network model, and the training can be difficult. It does not is not guaranteed to work well. We have seen how in practice you fix the hyperparameter of the network. And so for the designed sequences, what we have collaboration, for example, with the group of Ramaranganathan, which is able from a given sequence to synthesize the corresponding protein and to experimentally validate the functionality of this protein. And this is an experimental validation on inverse Potts model. We have not yet experimental validation of designed sequences with RBM. So this is the end.
Comments?
So if I understand correctly, you calculate the probability distribution and then you do Monte Carlo sampling to get the new sample.
Yes.
But if I go back and see the training part, I need to know the probability distribution. Then only I can compare whether what is if RBM is doing things correctly or not.
From the sorry, say it again. From your initial data.
So I have some initial data
Yeah.
and I have some probability distribution. I put that initial data into RBM and see
Yes,
what probability
exactly.
distribution is coming, and
Yeah.
then I compare it with actual probability distribution, and then I see whether they are matching or not. If they are
And what
matching,
are
it
the
means
actual-
RBM has trained.
You have the problem that, if, for example, on protein data, you don't know the real probability distribution.
Yeah, exactly. If I don't know the probability distribution, how would I know that whether RBM has trained properly or not?
With this cross-validation. So you compare the probability distribution of your data sets you have used to train the model with another part of the data sets, which is your test sets. So this you have not used to train the model, and you want the two probability distribution be the same,
Okay, so
or more or less as close as possible. Okay, so this is what is called a cross-validation. It's done only of part of data that you have not used for the training set.
And what is generally the segregation between the training part and the cross-validation part?
Sorry?
What is the segregation, like how much are you taking for a...
Normally it's like 10% of the data set for the test set and 90% for the train set. This is what usually is done in machine learning, or 20% and 80%.
Okay. And one more point. So when you are generating your protein sequence, that come, I don't know how that works, but I'm assuming that it has amino acids, so you are giving some kind of numbering to that.
You are, yeah.
But as far as I know, that RBM generally takes binary data.
Yeah, these are POTS variable. Yeah. Yeah, POTS models, on each variable, you have 20 possible states or color, which correspond, for example, to the different letters you can have. So it's like a generalization of IC model, which is for binary variable.
So just a question, when you do this RBM on MNIST or on images in general, you can use also a convolutional structure for the weights?
On MNIST, yeah?
You can use a convolutional structure. I mean, here I think that you are using a fully connected network. So you...
No, for the weights, I were using RBM.
Yes, but I mean, you have a layer with the visible variables which correspond to the pixels of the pictures. And in principle, you could use some convolutional structure in order to have some weight sharing connecting to the hidden variables.
Yeah.
And this is what you do for when you use convolutional networks. I guess you can do the same here also with RBM, or you just use fully connected weights?
No, I use weights which are, they are fully connected in the sense that each unit is connected to all the,
and all weight, each single weight is an independent value that you have to learn.
Yes.
So you don't have weight sharing. You don't have the same weight that is used for different.
Yeah, no, we don't use convolution network.
But in principle, is it possible to do this? You can enforce this translation invariance, right?
Yeah.
Okay. And the second question is, I didn't understand. Do you have some guarantees that these RBM actually are learning distribution?
So what we have done for this, we have tested on many artificial data for which we know the ground truth. For example, on POTS model, data trained from POTS model, and for the protein on model of proteins which are called the lattice proteins, which we use to generate samples, and then so we know the real probability distribution and we can compare with
so,
the one
for
obtained.
this kind of lattice models, Ising models, you can use this
Yeah,
because indeed you have a very similar structure in the model.
exactly.
But if you try to use RBM to learn a complex dataset like CFAR or any real images dataset, do you have actually any guarantee that in principle, at least with an infinite number of hidden variables, you are learning the distribution?
You are guaranteed in the sense that, and it's always the same question, your log likelihood on the test sets reproduce well the log likelihood on the trained sets. And you can calculate the value of log likelihood and compare, for example, with other methods and see that you obtain things which are very comparable.
But so you're saying that with an RBM, you can actually learn any possible distribution on any kind of manifold, at least if you have enough hidden variables?
If you can fix well your hyperparameter, and if you have enough data to well learn.
Of course, yes. Okay.
Talking about hyperparameters,
Mm-hmm.
why is it that the protein thing?
Yes.
So why is it in this protein example that you couldn't disentangle group 2 and group 3?
Yeah. So this is because
if you add more
we
unit, it doesn't change?
No. So we think that indeed they are not. So this separation can be also a little bit artificial. So probably one possible explanation that they are not really separated. So they are really a single cluster. They are not two different clusters.
Anybody else?
Training algorithm. I mean, you need, because you basically learn by gradient descent or something, and you need to estimate the gradient by some kind of Monte Carlo. So it's a painful process. Let's say.
So
if you do something much a raw approximation of the gradient, are the results strongly dependent on this?
So what we have used is the stochastic gradient descent method, which is in some sense the most stable and simple for machine learning. So we have not trained other methods for the moment. But this is really a very, in some sense, as it has been shown by the talk yesterday, it's really the one method which is really straightforward and simple. And in the case of RBM, this corresponds to the moment matching between the moments estimated by your probability distribution and the empirical moments.
Yes, but you need to compute the gradient by some kind of Monte Carlo.
And then
this is the part that is very costly.
Yeah. It's time consuming, but it's quite stable. Also on Boltzmann machine, we have tried different kind of approximation, but the Monte Carlo learning is one like really straightforward symbol methods, which then works well, even for Boltzmann machine.
Okay, so I guess it is getting a little late, so let's stop here and thank the speaker again.